Hello, my name is Hannah Bailey, and I'm a PhD student working under Dr. Hans Stein at the University of Illinois. Our lab focuses on determining values for digestibility of energy and nutrients, and in recent years we have been conducting research to determine digestible indispensable amino acid scores, abbreviated DIAS, for a variety of human foods. We specifically conducted this experiment to determine the protein digestibility corrected amino acid score, abbreviated PDCAS, and the DIAS for raw and roasted pistachio nuts in growing pigs. The second objective of this experiment was to assess the effect of roasting on these measurements of protein quality. The DIAS and PDCAS methodologies were developed by the Food and Agriculture Organization in 1989 and 2011, respectively. DIAS has been the approved successor to PDCAS for measuring protein quality. However, until more research is conducted to determine DIAS for human foods, PDCAS will be used. Pistachio nuts are edible nuts thought to be originally native to Iran, Afghanistan, and Central Asia. Pistachio nuts can be consumed in raw or roasted form, and compared with other nuts, pistachios have among the least lipid and greatest protein concentration. However, heat processing can affect amino acid bioavailability and therefore protein quality. To our knowledge, the quality of protein and the effect of roasting on protein quality of pistachio nuts have not been studied. Two forms of pistachio nuts were evaluated, raw and roasted. The pistachio nuts were roasted to a temperature of 115 degrees Celsius, which took approximately 30 minutes. For this experiment, 12 ileocannulated barrels with an initial body weight of 60.9 kilograms were used. The experimental design was a two-period switchback design with 3 diets using each ingredient as a sole source of protein and amino acids, plus a nitrogen-free diet to determine basal endogenous losses of protein and amino acids. There were 2 periods, and each period was 9 days, with fecal collection on day 6 and 7 and ileal digestive collection on day 8 and 9. A nitrogen to protein conversion factor of 6.25 was used in all protein calculations. PDCAS and DIAS were calculated at the conclusion of the experiment for preschool children 2 to 5 years of age, and for children older than 3 years, adolescents, and adults, respectively. Moving to the results, the crude protein content was greater in raw pistachio nuts, 27.1%, compared with roasted pistachio nuts, 25.1%, using a nitrogen to protein conversion factor of 6.25 as mentioned earlier. The results for standardized ileal digestibility, or SID, of some indispensable amino acids are shown in the top bar graph on the right of this slide. The orange bar represents raw pistachio nuts, and the blue bar represents roasted pistachio nuts. These colors also match the bottom bar graph on the right, showing the PDCAS and DIAS results. The SID of protein and all indispensable amino acids except arginine and phenylalanine, not shown on this slide, was greater in raw than in roasted pistachio nuts. For PDCAS and DIAS shown on the bottom right bar graph, the first limiting amino acid, which determines the PDCAS and DIAS value, for both raw and roasted pistachio nuts when compared with human amino acid requirements for the specific age group, was threonine for PDCAS and lysine for DIAS. The PDCAS for roasted pistachio nuts was 81, which was greater than the PDCAS for raw pistachio nuts, 73. In contrast, the DIAS for roasted pistachio nuts was 83, and less than the DIAS for raw pistachio nuts, which was 86. This illustrates that DIAS, which is determined via standardized ileal digestibility, more accurately determines the bioavailability of heat-damaged amino acids compared with PDCAS, which is determined via standardized fecal digestibility. In conclusion, raw and roasted pistachio nuts can be considered good quality protein sources with DIAS greater than 75 for children older than 3 years, adolescents, and adults. However, roasting pistachio nuts may decrease ileal digestibility of amino acids and DIAS, whereas PDCAS was not reduced. Therefore, PDCAS is unable to account for decreased amino acid digestibility due to heat damage from roasting nuts.